In this video, we're going to revisit the path that sympathetic fibers take in relationship to the spinal cord, to the sympathetic ganglia, and reaching their target. And then we're going to introduce the neurotransmitters that we see in the autonomic nervous system. So let's revisit this. In the past video, we talked briefly about sympathetic fibers starting off in the lateral horn. That's where their cell bodies are. And remember, these are the preganglionic fibers. They travel in the anterior root with the other types of motor information. They join the spinal nerve, which of course is a mixed nerve at this point because it carries both motor and sensory. And then the sympathetic fibers exit quite quickly from the spinal nerve to enter the ganglia, the chain ganglia. Now remember the concept that we need a pre-ganglionic fiber and a post-ganglionic fiber. We need both these neurons to go from the spinal cord to the target. And once again, the options are to get this synapse completed is that the simplest thing is that the fiber comes off. The sympathetic fiber then takes the white ramus, because it's myelinated fiber, takes the white ramus into the chain. It can synapse, so that's the end of the preganglionic, and the postganglionic starts, and the postganglionic is unmyelinated, so it's now in what we call the gray ramus, and then it rejoins the spinal cord and heads off. That's one option for these sympathetic fibers. Now, some sympathetic fibers have to head up higher than where they came off the spinal cord. So in that case, they can come off, they can take the white ramus, enter the sympathetic chain, they can travel up to the appropriate level that they need to exit the chain from, synapse, and then the postganglionic, once again, take the gray ramus and follow a higher spinal nerve to the target. The same thing, what if it has to go lower level? It comes in, it can travel to a lower level, synapse, and head off. Now there's one more option, and this option involves not synapsing in the sympathetic chain. Now, but do you have to synapse? Yeah, there's a golden rule that if you're a sympathetic fiber or parasympathetic, you need a preganglionic and a postganglionic. So what are you going to do if you don't synapse in the chain? Well, you come along, you do enter the chain, you use the chain as a route, but you don't synapse, and then you actually leave the chain without synapsing, which means you're going to have a ganglia somewhere outside the sympathetic chain. So something like the collateral ganglia. And you synapse there, and then the postsynaptic heads to its target. So there's the various options when it comes to sympathetic fibers. You have to have a presynaptic, you have to have a postsynaptic, you have to synapse. Many of them synapse in the chain, either at the level they came off at, or they go higher, then synapse and leave. They go lower, synapse and leave, or some of them pass through and then have their own ganglia on the way to the target. And these we refer to as the splanchnic nerves, a collection. Okay, so let's start talking a bit about the neurotransmitters. And of course, since they're neurons, they use chemical means to talk to each other. And we have to start looking at not only the neurotransmitters, but the receptors. And that's an important thing to remember is it's not necessarily the chemical that you release as a neuron. It's how the cell that's going to receive the chemical interprets it. So it's, as I said, it's not just about the neurotransmitters, but it's also about the receptors that are on, say, the postsynaptic neuron or postganglionic neuron as to how it's going to interpret the message. So let's have a look. So when we're talking about sympathetic neurotransmitters, of course, one big category is ACH, acetylcholine. And it's an excitatory synapse, and we see ACH being used between the preganglionic neurons and the postganglionic neurons. Right? Okay, so we have a little drawing down here. Here's the preganglionic. There's the postganglionic. So between the preganglionic and postganglionic, what's released? ACH is the one that we see being released. Now, how about for the postganglionic? We're going to see that there's some options. Now, just a reminder, one neuron doesn't have all these neurotransmitters. So if you're a neuron, you only have one of these in your pathway or as part of you, right? And you don't change day to day. You are what you are. So depending on the post-ganglionic neuron, they either release ACH, epinephrine, or norepinephrine at the region of the target that they're aiming for. So at the synapse of the target cells, as I just said, it's either ACH, epinephrine, also known as adrenaline, norepinephrine, okay, also known as noradrenaline. Now, which one is used most often? It is noradrenaline is the most common one between preganglionic axons and their targets. Now, this could be broken down even more because we just finished talking about the neurotransmitters. Let's start talking about the receptors. And when we start talking about the receptors, let's start talking about the adrenergic receptors. Now, before we go too far, think about adrenergic. Remember, the old name for epinephrine is adrenaline because it was first identifying the adrenal gland. So adrenergic, adrenaline, epinephrine, norepinephrine, all those names should really link in your brain. Now, when we start to look at what we call the adrenergic receptors, these are the ones that bind epinephrine or norepinephrine. And there's two main classes that we're going to look at. One are the alpha-1 receptors, and the other one are the alpha-2 receptors. So what's the difference? The difference is where they are and sort of what effect they'll have at the target. So where do I find the alpha-1 receptors? They're in the plasma membranes of smooth muscle cells. And of course, smooth muscle cells are in many types of organs, including things like skin and your blood vessels, so they can have an effect on the functioning of those. Your GI tract, your kidneys, the little muscles that make your hair stand up, the erector pillae muscles in the dermis, and certain parts of the genital urinary tract also have the alpha-1 receptors. And then we have the other group, which are the alpha-2 receptors. And these we find in the plasma membranes of preganglionic sympathetic neurons instead of being in the peripheral target cells. And we'll come back and kind of talk about why that would be. Now, let's kind of have a look at some of the other subtypes that we have out there. Okay, so we just talked about the adrenergic receptors, and we talked about the alpha-1s and the alpha-2s. And now let's talk about the beta class. So we have beta-1s, beta-2s, and beta-3s. And these are still an adrenergic receptor class. So alphas and betas fall into the same category. So the beta-1 receptors, where do I find them? In cardiac muscle cells and some parts of the kidneys and adipose cells. The beta-2s in plasma membrane of smooth muscle cells that I find in the bronchioles, which are, of course, one of the most important parts of the respiratory tract. And when we do the respiratory tract, it's going to make much more sense as to why they're there, because it helps to change the diameter of the blood vessel, which changes the resistance of airflow. Also in the walls of the urinary bladder, skeletal muscle fibers, liver, pancreas, and salivary glands. So you can see they're quite wide-ranging. And then we have the beta-3s, which are primarily in adipose cells and smooth muscle cells of the GI tract, walls of the GI tract. Now, Let's have a look at the cholinergic. And of course, if you just ponder it for long enough, you realize cholinergic kind of sounds like acetylcholine, right? Which is where the reference comes from. So cholinergic receptors bind to acetylcholine. And we have two types, the muscarinic receptors and the nicotinic category. So the muscarinic receptors we find in sweat glands in the skin, that's the predominant place, and for the nicotinic, in the membranes of all postganglionic neurons within the sympathetic ganglia and the adrenal medulla are really where we find those. So let's just talk a bit about the alpha-2 receptors, because if you sort of were paying attention and start thinking about the list, you notice most of these are about talking about target cells, but there was one, the alpha-2 receptor, which is different than the other adrenergic ones. And this has a role in terms of the action potential in the preganglionic neuron. Okay. Usually, the action potential in the preganglionic neuron leads to ACH release, which we've talked about, and then goes on and stimulates the postganglionic neuron. But if norepinephrine is around, it can bind onto the alpha-2 receptors. Now, we're going to see where the alpha-2 receptors are. They're not on the postganglionic neuron. They're at the axon terminal of the preganglionic, and it leads to hyperpolarization. And this will slow or even stop an action potential from influencing the preganglionic to release neurotransmitters to affect the postganglionic. And why does it exist? Well, it's part of a feedback loop that the preganglionic neuron activity is reduced or really shut down to prevent the sympathetic output from being too excessive. And let's have a look. So here's where we find them. Here are the alpha-2 receptors. So we're looking at a preganglionic neuron up here. And here are the alpha-2 receptors. So if norepinephrine binds onto these, as we can see has happened over on this side, that the receptors are now interacting with norepinephrine, what that does is basically prevents or really reduces the chance of the release of those neurotransmitters. Therefore, the postganglionic neuron does not get stimulated because I end up hyperpolarizing the terminal end of the preganglionic. So the postganglionic never receives its neurotransmitters, and therefore there's no activity there. So this has a big role when it comes to sort of limiting the activity of the sympathetic fibers. Okay, we're going to ease out there, and the next one we're going to come back and start looking at, well, what effects does this have on different types of target cells?